All right, hi guys. Here I'm going to lecture just real quick on introducing you to the respiration lab. I'm going fast and you're going to read about all this and watch it in the videos. So this is my second time. The first time I forgot to turn on the microphone, so here we go again. All right, so we're about to do the lab on cell respiration, which is we're going to measure the rate of respiration. And we're going to do it with peas, germinating peas versus dormant peas, which are seeds. So before we get into this a little bit, here's our equation of respiration, right? And how do you measure the rate? Well, we could measure the consumption of any reactant. Okay, so this is for any reaction. If you on the AP exam, it doesn't matter if it's the respiration or photosynthesis, or they just give you some random equation that's run by an enzyme or something and asking you, how do you measure the rate of this reaction? This concept applies. You have to, if you have a way to measure one of the reactants and you can measure its disappearance or its consumption, that would tell you the rate. Or if you have a way to measure kind of the appearance of products, how fast do the products appear? That would measure the rate of the reaction. Or sometimes, a lot of times, especially if it's sort of an exergonic reaction that's giving off heat, like respiration is, you can sometimes measure heat as a way to tell how much it's going. Okay, so the faster this is going, the more heat's released, or the more glucose is consumed, or the more carbon dioxide is produced. So we could measure it in any of these ways. The way that our method, it just depends on what method do you have to measure one of those things. We're going to measure the rate of oxygen consumption. Okay. So the amount of oxygen that is consumed per minute is going to be our unit. And that's how we're going to measure the rate of respiration in these organisms. We're going to use peas. Remember, peas, I know peas are plants, but remember plants do respiration too. So that's the first thing to understand. Okay, that's what we're measuring, the disappearance of oxygen. Now I'm going to go into, we're going to use a setup we call a respirometer. And it's all written here for you, but just kind of listen and I'm going to talk about it. It's just basically a glass vial with a stopper, and you put your organism in there and it breathes or it respires. Okay, and it's going to use up oxygen. And as it uses up oxygen, that's going to lower the pressure in there. It creates like a vacuum. I'm going to explain all this. And originally this pipette is open to the air, but when we start the experiment, we submerge it in water, okay? And so that as oxygen is consumed in here by the organism, it lowers the pressure in here, creates like a vacuum, and then water is going to get drawn in, kind of sucked in from high to low pressure. And if we have a pipette here with lines on it, we can measure how much water comes in is a direct relationship or directly telling us how much oxygen is being consumed. Okay, so that's our method. We have to create this respirometer, and which is kind of a closed system. And now we're going to have to get rid of carbon dioxide, which I'll talk about in a minute, because there's a gas being produced and a gas being used. Okay, so we have oxygen being used, but we also have carbon dioxide being produced, and I'll get to that here in our procedure. But all of this is based on, you have to remember the gas law back from, hopefully you had this in chemistry class, PV equals nRT, okay? When we're dealing with gases, remember gases take up any container, whatever container they're in, they spread out to that container. And what these things stand for, P is pressure, V is volume of the gas, and that equals N, which is the number of molecules of the gas. R is the gas constant, so that's just a number that's going to stay fixed. And T is temperature. And so I don't want to get too into the chemistry of it, but if you remember in an equation, in any equation, forget what it is, anything that is on opposite sides of an equation is going to have a direct relationship. As one increases, the other will increase, okay? Assuming the other things are constant, okay? Anything that's on the same side of an equation is going to have an inverse relationship, okay? So if I keep all of these things constant, and I decrease the pressure, that's going to make an increase in that means the volume is increased, or vice versa. Okay, so that's just general equation things here. So remember, we're talking about, a gas in this little respirometer. So just to kind of give you an idea here. So if I'm just considering, I have the P and the N in red, let's assume volume and temperature are constant, okay? Which we're going to try to, the temperature, these two things, let me change my color here. These two things, we're going to try to, in the lab, we're going to try to keep these constant, okay? Okay, so this, we're going to have to try to, the temperature, we're going to try to keep constant. We're going to try to keep that the same. We're going to put our respirometer in a water bath with a thermometer and try to make sure it doesn't change. But it might change, and so we're going to have to account for that a little bit. The gas constant, though, is fixed. That's just a number. That really has kind of nothing to do with understanding this right now. So consider that R and T are the same, stay the same, and the volume stays the same. If I'm just talking about the relationship between pressure and the number of molecules, if the number of molecules rises, if I have more molecules, and it's in the same volume, it's in the same space, what's going to happen to the pressure? So if I try to cram more molecules, what's going to happen to the pressure? Well, the pressure is going to increase as well. Okay, and so as N rises, P will also increase. Okay, if you think about that logically, like I just explained to you, or just remember they're on the opposite sides of the equation, so they're going to have a direct relationship, because on opposite sides of an equation of the equal sign. So that's going to be true of any two things that I pick that are on opposite sides are going to, as if one increases, the other increases also, okay? Assuming the other things are constant, okay? You can't be changing everything at once, then it's kind of a mess. So then, same concept, if N decreases, then P will decrease, okay? So if I have less molecules of a gas in a fixed volume space, then they will be under less pressure, okay? Assuming the temperature is constant. But we're going to see if we change the temperature. And remember, this whole lab is kind of based on the concept that water or gases or anything will move from areas of high pressure to low pressure. So the P, the pressure, is really going to be kind of what we're seeing here. Whatever affects pressure, if pressure decreases, water's going to move in. And so as oxygen gets used up and N goes down in that respirometer, the pressure will go down and water will move in. But if temperature were to not stay the same, we're going to try to. If temperature were to fluctuate, that's also going to affect the pressure. And so we have to have a control group that will measure that. Okay. But just here's another example, just kind of understanding the gas law. Well, this is really the one that we're going to do in the lab. Okay, this is what the lab is based on, right? This concept here. But just an idea, if I were to keep all of these constant, so the temperature is constant, I have the same number of molecules of the gas. How are P and V related? If P increases, then V will decrease. As if P decreases, V will increase. If V increases, P. Basically, you see they have what we call like an inverse relationship. Anything that's on the same side of an equation. All right. So that's just understanding the gas law and just general, like algebra, I guess. But again, understanding this lab is just kind of understanding that the number of molecules of oxygen in the container, if that changes, that's going to change the pressure accordingly. And that's how we're going to measure. Then water's going to come in accordingly, and that's how we're going to measure it, okay? And so this respirometer here, again, it's just, let me just kind of draw, it's just kind of like we have a pipette with an opening, okay? and that's attached to kind of a glass vial, okay? And so what we have here is we have sort of a closed system. And it's closed except for the opening of the pipette, okay? Which is the whole idea. And we're going to put our organism in there that we want to measure the rate of respiration. I'll just put a human in there. Pretend we had a giant respirometer. Or whatever, down here they have a cricket, and we're going to put peas. But you put the organism in there, and pretend this is closed for a minute, because as soon as we drop it into the water, okay, air can't go in and out, okay? So it'll be closed when we drop this in the water. And consider, we have our PV equals nRT in here. Okay, and remember, we're trying to keep everything constant, except that as the organism respires, okay, so we have all this oxygen gas in here in a fixed kind of space. It'll take up space, okay? That's the gas we're dealing with, okay? And then, so as the organism respires, whatever it is, respires, it's taking in O2, so the O2 is going to decrease in that fixed space. What that means is that the N decreases, right? The number of molecules of the gas that we're dealing with decreases. And as N decreases, remember, we're just dealing with N and P, okay? We're keeping the volume the same because it's in this fixed kind of space with a fixed amount of volume. R is the same, it's just a gas constant. And we're trying to keep the temperature the same. You'll notice this is in a water bath that we're going to try to keep it the same as the experiment goes. So as N decreases, what that means is that the pressure in here is going to decrease, okay? As the pressure decreases, think of it like there's a vacuum. It almost creates a vacuum inside, okay? The inside is going to have a lower pressure than the outside. Okay, so if we now have a lower pressure inside here than we do, and we have a higher pressure outside here, what's going to happen is the water is going to get sucked in. Okay, so water will move in from high to low pressure, from high P to low P. That's essentially how it works. Okay, so as an organism respires, the number of oxygen molecules goes down, the pressure in here then decreases, and that sucks water in, and we have sort of lines on here, and we can measure how much water goes in over time, and count how much oxygen was consumed then per minute, I'm sorry. Now, all of that, some of you are smart enough to think, but wait a second, there's two gases here, okay? And what really is happening, we have sort of a one-to-one relationship, right? Like for every oxygen gas molecule that is consumed, we have a gas molecule being produced. So in reality, if we weren't just considering oxygen, N would not change as an organism respires. It would take in oxygen, but it would give off CO2. And N wouldn't change, and pressure wouldn't change, and none of this would work, okay? This method wouldn't work. However, we have a way to get rid of the carbon dioxide gas as the organism breathes it. Okay, now it is going to breathe out carbon dioxide. Well, it's going to be inside the thing, but as the carbon dioxide gas is breathed out, we're going to see that we put this KOH in here, and this is a molecule that will have a chemical reaction with KOH, potassium hydroxide, has a reaction with carbon dioxide, which is a gas. KOH is a liquid, by the way, which doesn't really matter. And then that's going to react to give us this molecule you see over here, which you don't need to memorize it, but what's important about this is that it's a solid. It's a solid precipitate. So we're taking carbon dioxide as a gas and turning it into a solid. This takes up a lot of space, a gas. A solid precipitate takes up hardly any space or volume in that thing, in that respirometer. It's actually like, think of it like sand crystals. So I took a gas that would take up the whole container, the volume of the container, and I turned it into like a sand crystal. Okay, so that's not going to take up space and play a role in the pressure in there. So it's like we're taking it out of the equation. And so you can see here, we put KOH in it on a cotton ball in the respirometer, and it'll react with the gas in there. And you can see here, as the thing breathes out carbon dioxide, it reacts with the KOH. It forms these little, there'll be little crystals. You won't even see them. They're so small. But that just basically takes carbon dioxide gas out of the equation here, literally and figuratively, so that our method will work, that as oxygen's consumed, the pressure inside will decrease due to the gas law. So that's the method we're going to use. And you're going to read all about that and kind of see how they put this together. So there's my KOH, and then I have like another layer of cotton to protect the organism from KOH. This whole thing has to be sealed. Okay, we don't want water leaking in like there when the pressure changes, because there's no way to measure that. So the only place I want water to come in, well, I guess it would be more, is right here. Okay, the only place I want there to be an opening is right there, so that when the pressure does drop in here, I can measure how much water is coming in through the pipette. I don't want water leaking in anywhere else. All right, here's the setup. Again, like I said, instead of a cricket, we're going to set this up with peas. And you can see here, they go through this with you, but they put basically 25 peas. I think he might say 15, but whatever. So we put 25 germinating peas, okay, in the one vial or respirometer, and then we put 25 non-germinating peas, which essentially are seeds, okay, and they're dormant. So we haven't learned a lot about plants, and I don't want to get too into it, but a seed is basically, a seed is just a dormant plant. A seed, basically, you have a seed coat, which is kind of a hard structure. And then what's in there is a plant embryo. That's what a seed is. It's an embryo. If you ever eat a seed, you're eating a baby plant embryo. And then a whole bunch of food is put in there. And a lot of times it's starch. You know, it's food that's kind of stored in the seed, starch, which can be then turned into glucose, you know, which obviously is used for respiration to give ATP, okay. But in a dormant seed, okay, they're alive, okay. Seeds are alive. They're dormant, but they're alive. And so they have a very small respiration rate, just enough to keep it alive. It's not really doing much. It doesn't need a lot of ATP. It's just kind of maintaining itself alive. Whereas germinating peas, once this seed, you know, like a lot of times this happens in like the spring, okay, when there's a lot of water and kind of warm temperatures, water kind of soaks into the seed and the seed starts to germinate, okay, which just basically means it starts to sprout. And you see like it first gets bigger and then it sprouts a little root, and then a stem kind of comes out, and then eventually you grow a plant from it, okay? We all know a plant comes from a seed. So a germinating seed, pea, is a pea that's like you just planted it and it is about to sprout a new plant, okay? And so germinating seeds, whether it's peas or beans or whatever the lab may tell you, but we're doing peas, they're growing. So they need a lot of ATP, right, for energy, because growth takes a lot of ATP. And if they need a lot of ATP, that means they're going to have a higher cell respiration rate. So I basically am telling you what's going to happen in the lab. That's not, I think we could all guess that the germinating, growing peas are going to have a higher respiration rate than the dormant ones that are barely alive. But the method is what I want you to understand, and kind of the experimental setup here. So we're comparing, so that's one independent variable that we're doing. germinating versus non-germinating. That's one independent variable. We're going to compare germinating versus non-germinating. I don't know how I spelled that there, but independent variable is what I tried to write. Okay. Now, you may notice in this setup here, so we have 3 vials. We have germinating peas in here, and here's our cotton with our KOH and stuff down there. And here I have non-germinating peas, and then we have glass beads kind of in there with it. And then we have one with just glass beads. Okay, so this is going to be our control group with nothing alive in it, with just beads. And we need these beads in there, excuse me, to make sure that it's a controlled variable. Okay, just like this is, right? If I put 25 germinating peas in this vial, I put 25 non-germinating peas in the second vial. Okay, I have to keep that the same. That's a controlled variable, right? I can't put, 25 of these and only 10 of these and then compare their respiration rate. Like, obviously, it'd be different, okay? So, but the thing is that these are bigger. These take up more space. And so I have to add enough beads to make so that this volume is all the same, okay? So that all this is taking up the same amount of volume or space as that and that and that. That's another controlled variable. So that they all have the same amount of kind of air in there. So when you go to set it up, they use water displacement to figure out the volume that these peas take up. And then you have to add beads to these to make the same volume, and same thing here, okay? So it's going to ask you about like controlled variables in our setup. So there's 2 I just gave you. Remember, controlled variables are things we're keeping the same. We have the same number of peas in these 2 vials. These are our experimental vials. And we're making sure we have the same volume in that they take up the same amount of space in each vial by using the beads. Now, this is a control group, okay, like a whole group where I don't have any peas. And this is going to help me measure any changes that happen due to atmospheric pressure and temperature, right? Because we just said we know that because of PV equals nRT, we know that temperature, if the temperature changes, that can affect the pressure also and make water move in or out of this pipette. And we need to be able to see, okay, well, what if in this one with just the beads, I dunk this in the water and the water moves in by 0.2 milliliters? Okay. Well, that's not because respiration's going on. It's because maybe the temperature changed or the pressure changed. Atmospheric pressure just in the room or in the water bath. So if this one goes in, let's say, let's use simpler numbers. If this one goes in 2 milliliters and this one goes in 10, I have to subtract. We're going to have to subtract and do what we call the corrected difference. I know I'm talking fast and this is very sloppy, but the lab's going to go over all this, all the videos I'm going to have you watch. We're going to do what we call the corrected difference, where if this one moves 2 milliliters of water, that was not because of respiration. So I have to subtract 2 milliliters from whatever this one moved. If this one moved in 10, then only 8 of it was because of respiration, because 2 of it was what happened due to some other change in atmospheric pressure or temperature. Okay. And so we're going to subtract that from the other groups, and you're going to see that. So that's the control group. And these are my 2 experimental groups. Now that's one whole setup. And what you're going to see is we're going to then do that twice at 2 different temperatures. So we kind of have 2 independent variables. We have germinating versus non-germinating peas. Okay. We're going to compare those. And then we're going to do that at 2 different temperatures. Okay. So this is what the setup would look like if we actually did it. And you can see that we have, germinating peas versus non-germinating. So that's independent variable number 1. Right? But then we're also going to do this at 25 degrees Celsius and then another whole setup at 10. So this would be independent variable number 2. We're going to be comparing temperature as well. Okay? And so you guys are just going to watch a couple videos that show how to set this up and talk all about that again. But I just wanted to give you sort of an overview. And that